Welcome back to this next video in which we are discussing the auditory system. Okay, so in the previous video we saw that a sound is going to cause the oscillation of the tympanic membrane, which will cause an oscillation of the ossicular chain, which will then be transmitted into an oscillation within the cochlea. So we're going to get oscillation in the pressure of the scala vestibuli fluid, which will cause oscillation of the cochlear duct, which will then lead to oscillations in the pressure of the scala tympani, and then you'll get oscillations of the membrane of the round window. Okay, so we also saw how different frequency oscillations, different frequency sounds are going to lead to different portions of the cochlear duct actually oscillating best. We discussed how the basal portion of the cochlear duct, the basilar membrane there, is very tough, whereas the apical portion of the cochlear duct, the basilar membrane there, is far more malleable, and this means that low frequency sounds are more likely to cause oscillations in the apical portion of the cochlear duct, whereas high frequency sounds are going to cause oscillations in the basal portion of the cochlear duct. So different frequency sounds are going to cause different patterns of oscillation of the cochlear duct, and this is what we mean when we use this word tonotropic distribution of the cochlear duct. The cochlear duct can determine, or it can encode, different frequency sounds by which portions of it are going to oscillate. So each different frequency of sound will cause a different pattern of oscillation in the cochlear duct, and that's how we're actually going to be able to encode or determine what frequency sound we are actually listening to, because of the different pattern of oscillation of the cochlear duct that each different frequency sound will produce. Okay, so what are we going to turn on to in this video? We're going to turn on to how does the cochlear duct turn its oscillation into electrical signals, which can then go to the brain and be interpreted by the brain. Okay, so I need to introduce you then to a structure known as the organ of Corti, which is part of the cochlear duct that we haven't previously studied, but which is going to become really important now. Okay, so I'll colour everything to do with the organ of Corti then in blue. Okay, so the organ of Corti then, it's a structure that is sitting on the basilar membrane, and if I show it on this picture here, the organ of Corti will be in this position like so. So it's sitting on the basilar membrane, facing into the scala media where we have the endolymph. And again, this picture that I'm showing you here is obviously just a cross section, but the organ of Corti will go the entire length of the basilar membrane. So the entire length of the basilar membrane, you have the organ of Corti sitting on there. So the entire length of the cochlear duct has a portion of organ of Corti. And what I now want to discuss with you is what is the structure of this organ of Corti, because the organ of Corti is going to contain the structures which are going to transduce the auditory stimulus into an electrical stimulus. Okay, so here then is an enlarged picture of the organ of Corti here, but it's still a cross section. Okay, it's not a longitudinal picture, it's just a cross section. Okay, now the key cells that are in the organ of Corti are what are known as the hair cells, and you have two different types of hair cell. You have the inner hair cells and then the outer hair cells. Now the inner hair cells are the ones that face in towards the centre of the cochlea, so they're going to be on this side of the organ of Corti, and the outer hair cells, they're going to face out to the periphery of the cochlea, so they're going to be on this side of the organ of Corti. Okay, now, when we look at a cross-section of the organ of Corti, as we have here, all we're going to see is one inner hair cell, which I'll draw here, and these are the hairs, which we'll discuss in a lot more detail in a moment, and we're going to see three outer hair cells, which I'll draw like so. By the way, all of the rest of the tissue of the organ of Corti, all of the rest of this space here, that will just be supporting cells. The essential cells are these hair cells, so these are the ones we're going to focus on. We're not going to talk about the supporting cells, but that's what the rest of the tissue is, in case you're wondering. Okay, so what have we got here then? So this one, which I'll outline in green, that's an inner hair cell. And these three over here, which I'll outline in blue, these are outer hair cells. Okay, now, as I say, we are looking at a cross-section, which is why we're seeing so few of them. However, this continues on and on and on. So if I try and actually show a bit of longitudinal depth now, so let's try and show the organ of Corti going backwards. So of course the organ of Corti goes the entire way along the basilar membrane. I'm now trying to show that longitudinal dimension. The hair cells go back with the organ of Corti, like so. Now it's not just one giant cell, you've got loads of them. So here's one inner hair cell, then here's a second inner hair cell, here's a third inner hair cell, here's a fourth inner hair cell, a fifth inner hair cell, a sixth inner hair cell, and it goes on and on and on. You've got a great big row of inner hair cells all the way along the basilar membrane, all the way along the organ of Corti. Okay, so you've got a single file row of inner hair cells all the way along the basilar membrane, supported in this structure known as the organ of Corti. And it's the same for the outer hair cells, except that obviously this time you have 3 rows of outer hair cells rather than just 1 row, as you did with the inner hair cells. So here I'm trying to show the rows of outer hair cells. So there's the outer hair cells that are in the plane that I've actually gone through. Here then is the next row of outer hair cells, and on and on and on. A great big line, well, 3 lines of outer hair cells stretches all the way along the organ of Corti, all the way along the basilar membrane. So the entire basilar membrane, every section that you went to, so you could cut through at any section along here, you would always find 1 inner hair cell and 3 outer hair cells that you've managed to chop through, and they're in these massive great lines. Okay, and these are the key cells that are going to actually be able to transduce the movement of the cochlear duct, the oscillation of the cochlear duct into electrical signals. Right, okay, so let me now talk then about the tectorial membrane, I think is what we need to now discuss. So we have seen the organ of Corti. What we now need to discuss is how the organ of Corti's hair cells here are actually going to transduce the oscillation of the cochlear duct into electrical signals. And clearly, only the hair cells at the portions that will actually be oscillating will be activated to send electrical signals to the brain via neurons that will innervate them, but we'll see that later on. So what I'm trying to say is when the cochlear duct oscillates, it'll be the hair cells at the portions that actually oscillate for that frequency of sound that will be activated. The ones at portions that don't oscillate with that frequency of sound will not be activated. Okay, so the next thing that I want to introduce you to is the tectorial membrane. So we go back up to our picture of the scala media here. So there is another membrane that I need to tell you about, which is a membrane that's going to be inside the cochlear duct and which is going to sit on top of the organ of Corti. So I'll draw this like so. So it's attached here to the spiral ligament, and it stretches over the top of the tectorial membrane, over the top of the organ of Corti there. And we'll have this membrane shown here in pink. Okay, so that membrane, this is a very important membrane, it's called the tectorial membrane. And again, we are seeing a cross-section here, but the tectorial membrane will go the entire length of the cochlear duct. Okay, so I'll add it onto this picture down here. So the tectorial membrane would be going all the way along the cochlear duct, and I apologise for it sliding along. Okay, so it will be on top of the organ of Corti all the way along here, and in fact I might try and squeeze in the organ of Corti here. So in blue, this is supposed to represent the organ of Corti going all the way along the basilar membrane with the tectorial membrane on top of it all the way along. Right, so what's going to happen then? So we'll go back to the story. So when a sound comes in of a certain frequency, so let's say I've hit one of these bars of the xylophone, we're hearing a sound of a certain frequency. It will cause oscillation of the tympanic membrane and the ossicular chain at a certain frequency, the same frequency as the sound is. And then that will cause oscillation of the cochlear duct at the certain portions of the cochlear duct that are for that frequency. So I've pointed at the large bar here, so that would be a low frequency sound, so we'd be most likely to get most oscillation in the apical portion of the cochlear duct. So you'll get oscillation in the apical portion of the cochlear duct. Now, how is that going to actually cause a disturbance to these hair cells? Well, what's going to happen is that's going to move up and down, and you can see that what's going to happen is when the basilar membrane moves up and down, so here's the basilar membrane with the organ of Corti on top of it, that will cause the organ of Corti to move up and down, and that brushes the organ of Corti against the tectorial membrane. So I want to draw another picture of this here. So we'll have the organ of Corti here. Okay, we'll then have our inner hair cell there, and our 3 outer hair cells here, and let's add on their hairs here. And as you can see, the way I'm drawing the hairs, and this is very important, they have small hairs, and then it goes up to large hairs, and the small hairs are facing towards the inner portion, and the larger hairs are towards the outer portion, like so. So you can see there's a gradual increase in the length of the hairs. Okay, and I'll talk more about these hairs in just a moment. So here, this is the organ of Corti. Here is the basilar membrane on which the organ of Corti is sat. Let's just colour in the hair cells here. So we've got the inner hair cell in green there, and then the 3 outer hair cells in blue here. Okay, and then let's add on the tectorial membrane. So here, this can be the tectorial membrane here. Okay, so now if we imagine this moving upwards and downwards, which is what's going to happen at the portions which are actually going to oscillate with this frequency sound, what's going to happen is when the basilar membrane comes upwards, can you see that the hairs are going to be bent as they're pushed against the tectorial membrane? And in what direction do you think they're going to be bent? Well, I'll give you the answer. They're going to be bent in this direction, i.e. the small hairs are going to be bent towards the large hairs. And I'm going to give you the fancy word for the hairs of these hair cells. They are known as Stereocilia. So this is the fancy word for the hairs of these hair cells. So we can say that the small length Stereocilia are going to be bent towards the larger length Stereocilia when the basilar membrane moves upwards. And then of course as the basilar membrane comes downwards, everything will return to normal. But when the basilar membrane moves upwards, you get the Stereocilia bent in this way. Small Stereocilia towards the larger Stereocilia. And that's actually going to lead to changes in the electrical potential differences across the hair cells. This is how the hair cells detect the motion of the cochlear duct, by their Stereocilia being bent. So what I'm now going to do is draw a bigger picture of a hair cell. So here we are, and now I can actually show the Stereocilia. So they are finger-like projections coming off the cell, like so, that have a certain structure, which is why they're called Stereocilia. So they are a similar concept to, things like microvilli and cilia that you will have seen elsewhere in biology. But Stereocilia is the name for these ones that you have in the inner and outer hair cells here within the cochlear duct. And they will have a specific structure. I mean, the way that you actually create these things and maintain them, the proteins that are involved in creating them, will be specific to this type of projection in this way, so that's why it's known as a Stereocilia. Okay, so here is one of my hair cells, and this could be an inner hair cell or an outer hair cell. And we can see, once again, I'll remind you that the Stereocilia, there's a gradient. There's small ones at this end, which is the side facing towards the centre of the cochlea, and then there's larger ones towards the outer portion. Now, the Stereocilia are connected at their tips by structures called tip links. Okay, so these structures up here, these are called tip links, which is a reasonably sensible name, linkers of the tips of the Stereocilia. Okay, now, when the Stereocilia move in the direction towards the larger Stereocilia, so when the Stereocilia move in this direction, so the smaller Stereocilia moving towards the larger Stereocilia, what do you think is going to happen to those tip links? Okay, well, imagine this. The tip links are going to be stretched. Now, in fact, an easier way to see this is imagining the exact opposite. What if you move them in the opposite direction, i.e. large Stereocilia to the small Stereocilia? Then you can clearly see that's much more easy to imagine that the tip links will then become lax. Okay, they'll be less stretched, i.e. if I move this one in this direction, you can see that the tips are going to become closer, and therefore the tip lengths are going to get less stretch. Well, if you do it in the opposite direction, which is what the tectorial membrane is actually going to do when we move the basilar membrane upwards, you're going to get the tip lengths stretched. And this stretching of the tip lengths, by mechanisms that still are not completely understood, leads to the opening of potassium channels inside the membranes of the stereocilia. Okay, so what's going to happen is when the tip lengths get stretched, so drawing a little flow chart here, tip lengths get stretched, this is going to lead to potassium channels opening. Now, here's a really important concept that we need to discuss. These potassium channels opening is actually going to lead to depolarization of the electrical potential difference across the cell membranes of these hair cells because of the strange makeup of the endolymph. And let me just try and straighten this back up. So normally, in electrophysiology, when you open a potassium channel in the membrane of a cell, so let's say this is at some bog standard cell here, usually when you open a potassium channel in the cell membrane, this is going to result in hyperpolarization of the electrical potential difference across the cell membrane. And the reason is that normally potassium is very, very high inside the cytoplasm of the cell, and it's very, very low in the extracellular fluid outside the cytoplasm of the cell. Therefore, when you open a potassium channel, what ends up happening is potassium ions move from intracellular compartment to the extracellular compartment, and that's going to alter the electrical potential difference across the cell membrane. You see, potassium ions have a positive charge, so when I move potassium ions from the intracellular compartment to the extracellular compartment, that's going to alter the electrical potential intracellularly, and it's also going to alter the electrical potential extracellularly. Now, to draw a little graph of this, normally the electrical potential extracellularly is higher than the electrical potential intracellularly. So here are my abbreviations for electrical potential extracellularly and electrical potential intracellularly. So the capital E is for electrical potential, the subscript E here is for extracellularly, and this one is for intracellularly. So this is just meant to represent that this one is above this one. So normally if I move from the extracellular compartment to the intracellular compartment, I step down in electrical potential, and usually I step down by 70 millivolts. So normally the electrical potential difference across the cell membrane, the voltage across the cell membrane, is negative 70 millivolts or thereabouts, negative 65 to negative 70 millivolts. People quote the resting electrical potential difference differently, but it's around that sort of value. Now, when I open a potassium channel then, potassium goes out, that's going to lower the electrical potential intracellularly because you're losing positive charge, and positive charge raises the electrical potential of an area, so the electrical potential intracellularly drops down. Meanwhile, we're putting positive charge into the extracellular fluid, so the electrical potential extracellularly is going to step up. So what's now going to happen to the electrical potential difference across the cell membrane, how much I step down in electrical potential when I cross from extracellular to intracellular? It gets bigger, it gets more negative, i.e. normally you get a hyperpolarization of the electrical potential difference across the cell membrane. However, the endolymph, we know, has a very high potassium concentration, which is incredibly unusual for an extracellular fluid, and therefore when you open potassium channels in the membrane of the stereocilia, you actually get potassium coming in to the cytoplasm of the cell, and that does the exact opposite of what I've just talked you through. It's going to raise the electrical potential intracellularly, so this number will go up. It's going to lower the electrical potential extracellularly, so this number will go down because you're taking potassium ions from the extracellular compartment, and therefore the drop from extracellular to intracellular is lower, and that's called depolarization. Okay, so opening these potassium channels is going to depolarize the electrical potential difference across the cell membrane. Okay, so you get depolarization of the electrical potential difference across the cell membrane of the hair cells when the cochlear duct oscillates at that region where those hair cells are. And that's going to lead to release of neurotransmitter. You see, what these hair cells have at their basal portion down here is lots of neurotransmitter stored in vesicles. Okay, so I'll draw some of this here. And the neurotransmitter that we believe they use is glutamate. However, that's still a little bit controversial, but it's certainly an excitatory neurotransmitter, and of course the most famous, most commonly used neurotransmitter that's excitatory is glutamate, so it's a good guess. And what's going to happen is when we depolarize the electrical potential difference across the cell membrane, that will activate voltage-gated calcium channels, VGCCs. Calcium will then be allowed to come from the extracellular fluid into the intracellular fluid through these voltage-gated calcium channels. Remember, calcium is really high in the extracellular fluid compared to the intracellular fluid. It's ridiculously low inside the intracellular fluid, so whenever you open voltage-gated calcium channels, you're always going to get calcium coming in, and that calcium is then the signal for exocytosis to occur, so you're going to then release glutamate. So the overall message here is that the inner hair cells and the outer hair cells are going to start releasing glutamate in response to having their stereocilia bent in this way by being brushed up against the tectorial membrane when the basilar membrane moves upwards. So when the cochlear duct oscillates, these are going to start releasing glutamate. Now, that is the major function of the inner hair cells. The outer hair cells do this, however, their function in doing this is much less than the inner hair cells, and you'll understand that more when I actually talk about their innervation, which actually I think I'll do now. I'll come back to the other function of the outer hair cells later. Let me now talk about what this glutamate is going to do. So the inner hair cells and the outer hair cells, they're innervated by neurons, which are going to be excited by the glutamate and carry the electrical signal to the brain. And by the way, these hair cells, they do not conduct action potentials. Okay, they don't have the correct voltage-gated ion channels to conduct action potentials. All they are doing here is transducing this movement of these stereocilia into a change in the electrical potential difference across their cell membrane, and using that change in the electrical potential difference across their cell membrane to govern how much of a certain compound they are releasing. Okay, there's no action potential behavior going on here, it's just using the electrical potential difference across the cell membrane for signaling purposes. Okay, right. The glutamate that's released is then going to activate neurons, which will then use action potentials and send the signal to the brain. So let me now discuss the innervation of the inner hair cells and the outer hair cells. So I'll do this with this yellow pen here. So inner hair cells, they are innervated by a huge number of different neurons. Okay, they are innervated overall by probably around 20 different neurons. So I'm drawing these all on now, and this is each inner hair cell. Every single one of the inner hair cells all the way along the cochlear duct has around 20 different neurons, which all have their axons synapsing effectively with it, and when it releases glutamate, they're going to be excited to fire action potentials, so they're now going to be firing action potentials. In contrast, the outer hair cells here, they each have a single axon innervating them, so each of them has just one neuron innervating it. Okay, so this is what I mean when I say that the inner hair cells are the major ones which are about transducing this movement into an electrical signal. They are very densely innervated, and they're going to send the information to the brain via these neurons that are innervating them. The outer hair cells, you can see that they're much less important for that. They do it to a little extent, but what's their main function then if they're not on par with the inner hair cells? Well, their main function actually seems to be in potentiating the oscillation of the cochlear duct at the portions pertaining to that frequency. Now, how do they do this? Well, they're actually capable of contracting. So here we go. The outer hair cells are capable of contraction. And when will they contract? Well, they'll contract when the electrical potential difference across their cell membrane is depolarized. Now, when will their electrical potential difference be depolarized? Well, when the membrane has gone upwards. So we've talked about, with the sound, at certain portions of the cochlear duct, you're going to get oscillation of the basilar membrane upwards and downwards. And we've seen that when the basilar membrane deviates upwards, that's when the stereocilia are going to be bent and the hair cells are going to depolarize. now what's going to happen, I'm telling you, is that when they depolarize, the outer hair cells are actually going to contract. And can you imagine what will happen if they contract? Well, they'll shorten in length, and they're actually going to pull the basilar membrane upwards. Now, think what that will do. The basilar membrane was going up anyway when the hair cells become depolarized, but now you're going to get it pulled up further. Okay, so you're potentiating the oscillation at the certain portions of the cochlear duct that are for that frequency of sound. Then, when the basilar membrane comes back down, what will happen is these hair cells will repolarize, okay, so their depolarization will go away as their stereocilia return to the normal state, i.e. not bent over, and then they'll lengthen back out, and that relaxation will help the membrane go back down, so it will potentiate the membrane going back down. So the point is that these outer hair cells exaggerate or potentiate the oscillations at the certain portions of the cochlear duct for that frequency. Okay, so that we think is one of the major functions of the outer hair cells. So really it's the inner hair cells that majorly transduce the information into electrical signals. Okay, so the final thing that I want to go over in this video is where are the cell bodies of these neurons. At the moment I've just shown you the peripheral processes. So the neurons which innervate the hair cells, both the inner and outer hair cells, they are examples of bipolar neurons. And a simple picture of a bipolar neuron is like so. So this is a bipolar neuron. And all bipolar means is that they have two processes coming off them. And one of these processes is going to be the peripheral process, and this is the process that's going to go and innervate and synapse with the bottom of the hair cell. Okay, so this is the bit that's going to be stimulated by glutamate and cause an action potential to fire up it. And then they've also got another process, which is the central process, and this is the one that will then feed the action potential into the central nervous system, into the brain. Okay, so if they fire an action potential, it will come up the peripheral process along the cell body here, then along the central process, and it will then synapse onto some next neuron along a secondary neuron, and the information will be transmitted further. Okay, so these first order neurons that innervate the hair cells of the cochlea are going to be these bipolar neurons. So the question now comes, we've seen their peripheral processes, where are their cell bodies going to be? Well, their cell bodies are going to be in a structure known as the spiral ganglion, which is at the base of the spiral ligament. So what I just want to do is draw a little picture again of the modulus. So remember, we drew this cross-section long ago. So here's the modulus, and here are the spiral canals around the modulus. So we've taken a transverse cross-section through the cochlea, like so. Okay, now let me put on the spiral ligaments here. Okay, and we know that these peripheral processes are coming from the organs of Corti all the way around. That's the really important thing to note. The basilar membrane goes all the way along the cochlear duct. The organ of Corti goes all the way along the cochlear duct. You have these inner and outer hair cells all the way along the cochlear duct. All the way along the cochlear duct, every single one of the inner hair cells will have 20 bipolar neurons here innervating it, and each bipolar neuron will just innervate one inner hair cell, and each outer hair cell will have one bipolar neuron innervating it. All of these bipolar neurons have their cell bodies at the base of the spiral ligaments, so in these positions here. Okay, so their peripheral processes go off to the organ of Corti, and then their central processes all come into the centre of the modulus, and they'll all converge to form a massive great nerve here, and that nerve is called the cochlear nerve. So they're all converging to form, and I'll just make sure this is in view, yes it is, the cochlear nerve down here. And of course the cochlear nerve, now I'll move this up to here because I'm running out of space, the cochlear nerve will then fuse with the nerve coming from the vestibular system, which is called the vestibular nerve, to form a very famous nerve that every medical student knows the name of, which is the vestibulocochlear nerve, cranial nerve 8. And of course this will be happening on both sides, although we're just looking at the right hand side. So this is the vestibulocochlear nerve, and this portion up here is the vestibular nerve. Okay, so the vestibulocochlear nerve is cranial nerve 8. And of course we will follow the path of the vestibulocochlear nerve in the next video, and we'll see where the central processes of these bipolar neurons are actually going to end up and synapse. Okay, so this base then, at the base of the spiral ligament where you have all of these cell bodies of these bipolar neurons, this has a name. This is known as the spiral ganglion. So remember, a ganglion is a place outside of the central nervous system where you have the cell bodies of neurons present. And the reason this is called a spiral ganglion is because it does spiral around the modulus. Remember, the spiral ligament spirals around the modulus. It goes all the way around with the spiral canal, like that portion of a screw, okay, that you use to actually screw the screw in, okay. And now at the bottom of that spiral ligament, you're going to have cell bodies, neuron cell bodies everywhere along, and that's now called the spiral ganglion. So it's a ganglion that actually is in the shape of a spiral. So if I was to draw it out, it would be like this from the front, going round with the modulus. So it would be around the modulus, which is that cone at the centre. Okay, so that's the spiral ganglion. So all of these bipolar neurons, they have their cell bodies in the spiral ganglion, and they send their central processes into this forming auditory or cochlear nerve, and that's another name that you can use for the cochlear nerve. You can call it the auditory nerve. Okay, and the auditory nerve fuses with the vestibular nerve to form the vestibulocochlear nerve. So let's summarise what we have now seen. So the really important thing to remind you at this point is how we're going to encode frequency information and how we're going to encode amplitude information, the two parts of sound. Okay, so remember frequency first, because that's more complicated. Frequency is going to be encoded by which neurons are actually firing action potentials, because remember, different frequencies will cause a different pattern of oscillation in the cochlear duct. They'll cause different portions of the cochlear duct to oscillate, okay, and therefore you will get different activation of hair cells, because you'll get the hair cells at the portion that's actually oscillating being activated, and therefore you'll get those neurons that innervate those hair cells at that portion that is oscillating firing. So which neurons are actually firing encodes the frequency information. So we can say that frequency is encoded by spatial factors, i.e. which neurons are actually firing, the neurons from the portion of that innervate the portion of the cochlear duct that is actually oscillating. Okay, so different frequencies then will cause different neurons at different parts of the spiral ganglion to actually start firing, and that's the way in which we are encoding the frequency information. Amplitude information, well, that's simpler to understand. If we have a higher amplitude sound, a louder sound, we have bigger oscillations, we're going to get larger oscillations of the cochlear duct, and therefore we're going to get larger bending of the stereocilia. We're going to get bigger depolarizations of the hair cells at that portion that's oscillating larger, and we're therefore going to get more depolarization, more glutamate released, more frequent action potentials firing up the bipolar neurons. So the rapidity of action potential firing by the bipolar neurons at the portion, well, which are innervating the portion of the cochlear duct that is actually oscillating, encodes amplitude. So firing rate of the bipolar neurons encodes amplitude information. Okay, so that's how we encode the two pieces of information about sound. Which neurons are actually going to be activated encodes which frequency of sound we're looking at, because, as I say, there is this tonotropic map on the cochlear duct, and the firing rate of those neurons that have been activated encodes the amplitude of the oscillation, because the higher amplitude sounds will cause more glutamate to be released by the hair cells, and therefore more rapid firing by those neurons. Okay, so in the next video we'll have a look at the central auditory pathways. We'll have a look at where the vestibulocochlear nerve is going to enter the brainstem and how the information is going to be transmitted from there.